Alright guys, welcome back to Underground Science, and in this video we'll be looking at the part 3 of our immune system and lymphatic system. So we'll pick up from the part 2 video. So in the part 2 video we end off right here saying that we talked about all the fluids coming in, the pressure rising, and the flap-like valves and endothelial cells of the lymphatic vessels snapping shut. Alright? And we also said something here that got cut off is that we then go to another important, very important structure called our lymph node. All right. So now we're going to start talking about here and what happens here. All right. So this is our lymph node. Okay. So in our lymph node, we have all of these things coming in. Okay. So we have the fluid, right? That's coming in. So we'll draw some fluid here, right? We'll draw a little fluid here because we need most of the space to talk about the immune system activity that goes on. So one thing to know that in our lymph node, we're going to have most of our adaptive immune system taking place. All right. So most of our specific and adaptive immune system taking place, involving our cell-mediated response, so involving our T lymphocytes, our cell-mediated response, and involving our humoral response. All right, so and involving our humoral response, involving our B lymphocytes. All right. So what's going to happen here? So let's just straight up draw all these things here. So we're going to end up with our pathogen, right? So let's draw two of the pathogens. All right, then we're going to have our fluid, which we already drew. Then let's draw a dendritic cell. All right, just draw one dendritic cell here. Okay, so let's draw a dendritic cell here. And once it pretty much comes here, by the time it comes here, right around here, it's actually going to destroy the pathogen, because remember it had the pathogen phagocytose inside it. So it's going to use its own hydrolytic enzymes, destroy it, and antigen present it on a MHC2 complex. All right. So then we're going to have a MHC2 complex. Remember to go check out all these videos, right, that we've talked about all this already. And we're going to have part of that antigen of the pathogen presented. So it's a smaller peptide of this huge protein pathogen here. Okay. And again, these are color codes. So this is our dendritic cell. And now what we're going to have in our lymph node, guys. So in I believe it was our part one video, I think. We said that our lymph node was divided into a cortex and then a medulla. Our cortex involved most of the B cells and our medulla involved most of the T cells. Now for the purpose of getting the concept down, we're not going to draw the cortex and medulla. We're just going to draw all the T cells and B cells working here, right? But just know that we do have a cortex and medulla of our lymph node. All right, so let's go ahead and now draw a T cell and a B cell. All right, so first let's go and draw our B cell here. So what our B cell can do is that our B cell can actually remember. So first of all, let me just go and draw this. Try to draw in a different color. Let's just draw in straight up black, I guess, so we can differentiate between the colors. So it's going to be our B cell. All right, so what our B cell can do, remember, is from our B cell videos that for one, right, it has membrane-bound antibodies. So if we drew our membrane-bound antibodies, not with green, right, not with green. If we drew our membrane-bound antibodies, membrane-bound, let's just go and draw a couple here with our heavy and light chains, right? And so we have membrane-bound antibodies. On top of that, we know that our B cell can act as a professional antigen presenting cell. So it can actually take this virus, right? So not like that, sorry about that. It can actually take this virus or bacteria, whatever pathogen this is, it can take it in, break it apart using hydrolytic enzymes, and then it can actually present it on a MHC2 complex. Okay, so this is our MHC2 complex, and this is also our MHC2 complex. And it's going to take it and present it on a MHC2 complex. Okay, so what can happen next? We know from our previous videos is that now we can have T cells. So now let's go and draw our T cells that come into play here. So to be honest, we're actually kind of running out of colors, so I'm just going to straight up just start drawing with the color, right? So let's just go ahead and start drawing our T cells here. So our T cells will come along and help us, right? So since we're running out of colors, to differentiate, we'll just start labeling. So this is our B cell, and this is our red is going to be our T cell, all right? So our T lymphocyte. Remember, our B cell, and in fact, our B cell actually can bind this whole pathogen to the antibodies also, so we can show that too, right? So we have our pathogen bound into the membrane-bound antibodies of the B cell. All right, but anyway, so what our T cell can do, let's take this T cell for a moment and actually erase this pathogen here. So our T cell can remember, it can go up to our antigen and start the binding process. All right, so it's going to erase this and draw it closer to our antigen. That's bound to our MHC2 of our B cell. So our T cell, if it matches, right? So if our alpha and beta chain, right, match, if our alpha and beta chain of our receptor, of our T cell receptor, match the antigen or the complementary antigen binding spot on the MHC2, it will go ahead and bind, right? Successfully bind. But we know that's not enough. We know that we need another structure, another glycoprotein. All right, so we know we need another glycoprotein. So this glycoprotein is going to be, so it's going to fix this real quick. So our glycoprotein is actually going to go over here and bind to our MHC2 complex from our helper T cell. So in particular, right, this is going to be our CD4 glycoprotein. And in particular, this is going to be our helper T cell, because we know helper T cells are CD4 positive white blood cells because they have the CD4 glycoprotein that will bind to the MHC2. Now, once this happens, we're going to have some lymphokines and cytokines released by our helper T cell that will go ahead and act on the B cell, which will cause the B cell to start proliferating into memory B cells, right? So we'll just say MBC, and it'll start proliferating into effector B cells or plasma B cells, all right? So memory B cells and plasma B cells. So let's go and write that down so we don't confuse the abbreviation. So MBC stands for, in this video, right, in this case, memory B cells, and plasma B cells stands for, or that's what it stands for, right? Plasma B cells. So that's what we're going to see. And also, not only will our helper T cell release lymphokines to stimulate the proliferation of our B cells, but our B cells will also release some cytokines that will in turn go over to our helper T cells and stimulate the proliferation of our helper T cells into two other structures, right? Or into two other types of helper T cells, which are going to be memory helper T cells and so let's go and draw another arrow, and into effector helper T cells. Okay, so let's go ahead and write those abbreviations down. So MHT in this case will be memory helper T cells, and effector helper T cells, or EHT in this case, is going to be effector, right? Effector helper T cells, all right? And so our plasma B cells are then going to start to produce a bunch of antibodies against this specific pathogen. So the antibodies are going to have the same exact variable portion receptors as these membrane-bound antibodies do. So it can go to other places of the body where this specific pathogen is and opsonize it. All right. So then these plasma B cells are going to start to produce a bunch of free-floating antibodies. Okay, so a bunch of free-floating antibodies. And so these free-floating antibodies, so remember, we're saying that the plasma flow in this case is going this way. All right, so if it's going this way, we know lymph flow is one direction, our lymphatic system. So our membrane or our free floating antibodies will then go from our lymph node and continue on in our lymphatic system until it reaches, right, until all the lymph collects, right, mostly in two spaces, but this time we're just going to take an example of our thoracic duct. And then our thoracic duct will actually empty into the left. So actually just draw an arrow here. We'll empty into the left subclavian vein, and then a left subclavian vein will then empty into our superior vena cava, which we should know based on our cardiovascular system playlist, all right? And then it'll get pumped into circulation. Then what can happen is that these free-floating antibodies, once it gets pumped back into circulation, so eventually, right, this will get pumped out into circulation via the heart, and then will actually come back. So now it's going to be here, right? Because this is where it all started in the arterial side. Okay, so what's going to happen is that many of these free-floating antibodies made by the plasma B cells will then get pumped into circulation, come back to this place. So these are all inside the blood vessel. Will come back into this place and will actually squeeze through the endothelial cells of the blood vessel and enter the area of infection, right? Area of havoc, all right, through a mechanism. So it's going to squeeze through the endothelial cells through a mechanism called diapedesis. All right, and we'll see why our antibodies can squeeze through the endothelial lining through diapedesis is because we'll actually have a lot of histamine and heparin released from our mast cells, right? So these, and we'll talk about that in the next video, all right? So our part 4 video. So see you guys there, all right? So we'll actually pick up right from here. Okay, so see you guys in the next video.